I've got Zoom recording and I'm recording on my computer. So hopefully this will work out pretty well. We've got more people showing up. So we're going to give it a couple more minutes. To allow people an opportunity to pop in, and then we'll have a chat with Dr. Boulton. All right, so there we go. So everybody can see this really quickly. That's pretty cool. I worked like a whole 10 minutes on that. I'm just kidding.
Is that actually my hand? It could be.
That could be your hand.
Yeah. I don't think that is, but although I do have a magnifying glass very much like that one. So, yeah.
Oh, okay.
Excellent piece of work there.
Well, thank you. It's amazing what technology can do. So we have space for up to 100 people in this particular discussion. So if you would like to invite other people, please, I suppose, feel free. It was open to the public through Southwest. So they sent out an email and posted on their Facebook page. So that's pretty neat. Sorry.
Oh, that's fine. So we're approaching 5 minutes, and I think let's just initiate. What do you think?
Yes, we can go ahead and get started. Okay. So welcome to the Natural Science Seminar through the Natural Science Life Science Department. This is Dr. Ian Boulton. He has been with us around this time last year for our first National Science Seminar, where we connected 4 campuses that day. That was pretty awesome. So thank you so much, Dr. Boulton, for agreeing to come back and do another Q&A session.
Yeah, it's my pleasure to be here, and it's great that we have the technology now to easily do this kind of communication from my home here in the Netherlands across the Atlantic to you guys. And yeah, it's a pleasure to be here and to talk a little bit about pandemics and vaccines, because these are obviously key subjects that we're all focused on in one way or another at the moment. So should I just continue and do introduction and the short talk, Mel?
Absolutely. Why don't you give a little bit of background about what's your education and what you've been working on? I know I've shared with my students some of your work, and they're very, very impressed, particularly when we were talking about your Nature paper and the how you managed to find a compound that was reducing the efficiency, was it, or the efficacy of vaccines?
I've had a great deal of opportunity in my life, and also the focus to really drive that home as far as I could. So there are some downsides to that in terms of life decisions, but yeah, but I've been involved in this field for the last 25 years or so. Firstly, I'm not a physician, and I'm not qualified to give medical advice. So nothing that I say here today should be considered that. If you need medical advice, you should talk to your own physician licensed in your area. In addition, I'm speaking for myself here this evening. I'm not paid by or contributing to any corporate entity at present. My initial training was in medical microbiology at the University of Edinburgh, and subsequently biochemistry at the University of London, where I did my PhD. And subsequently, I've been involved in research and development of vaccines, biodrugs, and most recently transgenic cells for various conditions, including cancers and some infectious diseases in terms of the vaccines. Uh, a recent job I had was with Janssen or J&J on one of their COVID vaccines, and I've since moved on from that, having completed the job. Um, with that as a brief introduction to me and sort of the maybe the credibility that I can bring to the discussion, I wanted to move on and talk about pandemics and infectious disease and vaccines as an introduction, and also to drive for questions in the audience. And the last sort of half to two-thirds of the time will be devoted to that, because the questions are more important than the raw information that I can give you, but it might contextualize and stimulate some. Um, so, Miss Williams is okay. I'll just continue.
Feel free to go for it, Dr.
Yeah,
Boulton.
perfect. Thank you. So, to a large extent, infectious disease is a symptom of our success as a species. What do I mean by that? Uh, in order for an infection to rip through a population in the same way as we're seeing now, there has to be a sufficiently large and or sufficiently mobile population to sustain that disease spread. And since we started to live in expanded family groups, villages, and then towns, and then cities, and as we started to move around the world routinely, the the same effects that used to result in outbreaks in small local populations are now able to spread through tens of thousands, millions of people. Um, our species has succeeded in colonizing a lot of the planet and moving around effectively. Consequently, we have to deal with outbreaks of infectious disease, and an outbreak that is across essentially the entire population is a pandemic. For almost everyone in the room, this is the first experience of this kind of thing that we've had. But going back through history, there are accounts of disease outbreaks and pandemics going back for as long as we have been able to record them. Certainly in Europe, the Yersinia pestis bubonic plague pandemic um, devastated the population on several occasions during the Middle Ages and subsequently in the 17th century. That's one example. A more recent example is the influenza pandemic, which ripped through Northern Europe and America in the years around the First World War. And to put that into perspective, more people died as a result of influenza, that particular variant, than died in the entirety of the war to end all wars, the carnage of the First War. So, in terms of perspective, the impact of an infectious disease can be enormous and dramatic and devastating. And this has happened in the past, and we're still here to talk about it. So, we will get through this. There's no doubt about that. And although the coronavirus outbreak is distressing and is really alarming in terms of the number of people that have it and the people who are dying, the overall mortality rate seems to be around 1 to 2%, and the people who pass away are usually people who have are either elderly or have a comorbidity, another condition that makes it harder for them to either mount an effective immune response or deal with the symptoms. That said, although that number appears to be very small in a large population such as the 330 some million of the USA, that's an enormous number of people. Um, Another point that is important to reflect on is that we've never been in a better position to be able to respond to a public health challenge like this. So, within the last 12 months, a number of vaccines have been prepared not from nothing, but from the development stage to the clinical stage. And that's an extraordinary achievement and a really remarkable achievement, carried out by people trained in environments like most of the audience are training in, uh, and in labs like the ones I set up and sometimes run. Uh, so although there's a great deal of human pain and suffering associated with this pandemic, I'm very confident that we'll turn the corner collectively as vaccines are rolled out and administered. And although it's very tempting to ridicule the people who are either anti-vaccine or anti-mask, um, it's very easy to ridicule them as well. Using the data, we have to have a historical view of this as well. Every other time there's an actual crisis, an infectious disease challenge, and a public health emergency, there's a small population of people who cannot accept that that is the actual reality of the circumstance, and will do everything they can to reject that reality. And I think they're much more to be pitied than vilified. Although unfortunately they do damage in terms of a coherent response to a pandemic like this, which, to be clear, should be social isolation as much as possible, masking and hand sanitization as a general rule, and extreme care over people who are at high risk. And by that, I mean the elderly and people with immune dysfunction or people who are otherwise unwell. Um, so that's a very quick review of historical pandemics, our responses to them, the current situation. And I think at that point, rather than laboring any of the subject areas that I've dipped into, let's move to audience participation and see if there are any questions that we can unpack.
Let me unmute myself. Fantastic, Dr. Bolton. So we do have, thank you again for that rundown. I know there's been a lot of misconceptions and lots of resistance on vaccine resistance and mask resistance. And so it's really helpful to have you here to be able to to answer a lot of these questions that different people
Yeah,
have.
yeah.
So
What I forget sometimes is that the type of thought process and discussion and data driven analysis that I have to do as a professional scientist isn't always how other people think. And it's perfectly understandable to say, I'm not sick. Why would I have any intervention? So I completely get it. And that's why the
mm.
questions are so important.
I don't know if you're familiar with any of the work that St. Jude is doing on vaccines. I don't know if I know you're kind of
I've been a little bit focused.
a little bit focused. So somebody had a question about the Johnson and Johnson vaccine also being the same as the St. Jude vaccine. I'm not
I,
certain.
I, I don't know.
Not, that's
It's
okay.
an interesting and very effective vaccine based on what I've seen,
Mm,
and it's based on an adenoviral vector that can get into your cells but not cause infection. So your body responds like it would to a virus and makes a really powerful T cell and B cell response, which is
mm.
exactly what you need.
Awesome.
Yeah.
Anne asked a question. How is the vaccine that you worked on different from Pfizer and Moderna?
Yeah. To speak generally, a vaccine requires a means of delivering an antigen and a way that, and to be formulated in a way that stimulates a response. So, two of the vaccine, or one of the vaccines, is an antigen-based vaccine. One is an mRNA-based vaccine, and one is a viral vector vaccine. So there are three very distinct groups, but they all do the same thing, which is either deliver an antigen and stimulate an immune response in an antigen based vaccine, deliver a signal that allows an antigen to be expressed, as in the mRNA vaccine, or deliver a quote unquote disabled infectious agent able to deliver the same signal as in the J&J vaccine. So there are all different routes to get to the same point.
Um, Dr. Roy, Dr. Roy, I call him Dr. Roy because I always say his last name wrong, and I don't want to do that. Dr. Roy asked a question about the Johnson and Johnson vaccine. It
Mhm.
only needs one shot, but others need two.
Yeah.
If you could explain, please explain why.
Well, I would still recommend two. I've had two of that. Um, but from the clinical trial data that we see, one shot uh, is extremely effective. And I alluded to the way the way the antigen is presented in this case as being very close to the same infectious process as COVID uses. So as a result of that, the antigen processing stimulates both the T and B cell responses, and both of those together give you a much better shot at a protective immunity than steering heavily towards one or the other. So the B cell response is the antibody response. It's a protein-based soluble response that flags up invading organisms. And the T cell response is a cell-mediated one, which can actually kill cells that are infected. Um, and in terms of viral infection, both are very valuable.
All right. I had a question in my head and it just flew away for a second. Here it came back. Just kind of piggybacking a bit on that. There are some vaccines that elicit a stronger immune response than the virus itself.
Yeah.
Do we have any kind of data like that for COVID-19 for the
I'm
vaccine?
Not that I'm confident of, but typically a vaccine will give you a higher immune titer than a natural infection. But natural infection is not a single beast. It ranges from minimal to massive. So it's two different moving targets. So it's very difficult to answer fully.
Okay. Ashley Williams has a question.
Mhm.
How will vaccines slow the progression of spreading the virus/disease?
Okay. If you imagine a disease, an infectious disease, to be fire, and the human population to be a really dry forest. Um, the fire can spread through all the dry tinder. However, a vaccine damps down that tinder, making the overall vulnerable population smaller, meaning that if I have an infection, I can't spread it to more than one other person instead of spreading it to 5 other people. So the vaccine effectively takes individuals out of the vulnerable pool. They're protected. They cannot be seen by the virus. The virus can't spread through them to others. So the overall network of infection, one person infecting 2 others who infect another 2, breaks down. And all you need to do is sustain one person spreading to less than one. And eventually the numbers just drop way down. And I'm confident that, consistent with vaccine deployment in the US, which is really, really going well now, um, you'll begin to see that over the next 2 months in the USA.
Well, thank you for that. We have another question. Seems we have quite a few questions, so I'm
Yeah,
excited.
good.
Okay.
Yeah.
So Robert Driver asks, what are the differences that make the new strains of COVID-19 more transmissible than the original strain of the virus? And then there's a second half to that.
Mhm.
Are the concerns that further mutations might make our vaccines less effective?
The answers are one, I don't know, in terms of the difference between the new strains and the old strains. Um, What I am aware of is that the key vaccine antigen, the S or spike protein, is conserved in all of them. So the current vaccines remain effective. Am I concerned about vaccines becoming less effective as the virus changes? Yes. But that's true for all vaccines and all viruses and all pathogens because evolution is an ongoing process and has a wonderful ability to fill any available niche. Viruses are particularly interesting because they can change more quickly than other biological entities for various biochemical reasons. But yes, this is something that is a concern and is very much something that we're aware of. For instance, the flu vaccine changes seasonally as a function of flu virus changing as it moves through populations. And there's no reason to assume that COVID will be significantly different from that in a broad sense.
Okay. Well,
Mhm.
thank you. Brooke has a question. We have a question from Brooke.
Mhm.
What percent of the population of the world needs to be vaccinated for it to work? And how often does that frequency differ from type of vaccine?
That's a huge question.
That is a
And
big one.
yeah, it's not a question I think I can answer specifically, but I can answer it in a general sense. And I think what we're driving towards is the term that's used in being herd immunity. And what that means is in a population, there are an insufficient number of vulnerable people, people exposed to and at risk of the infection for it to sustain itself in an ongoing sense in that population. Uh, the precise numbers related to that vary depending upon the population and upon the condition. But we've essentially reached that situation with smallpox and in many places in the world with polio and some other conditions. A rough rule of thumb is 70%. But again, it varies depending upon the population and the condition involved.
All right. So we just got the questions coming in. We have a lot of engagement going on, which is
Yeah.
awesome. So that's cool.
I think
Um,
there was a second part to Brooke's question, which was how
yes.
often, which I'm decoding or interpreting as uh, how often do we need to get or will we need to get a COVID vaccine to remain protected?
Right.
Again, I don't know the answer to that question. Um, there's a word of caution here. When a new thing happens and lots of reports come in, the tendency is to give them all the same level of credence. So people are infected and then reinfected and they've had COVID multiple times. And we hear that, but I'm not confident of that being an actual thing that's established yet. And as more data comes in, that'll be clearer. But from the serology I've seen, the protective effects of at least the vaccine I've been working on are extended out to multiple months, and the studies are ongoing. So I have a good level of confidence that this won't necessarily be, excuse me, something that you need every month or 3 months or 6 months or even annually. However, there's a quite high likelihood that this will be added to a seasonal schedule of vaccines, especially for people who are either vulnerable themselves, like my elderly parents or my sister who has an immune condition, or people who work with folks who are highly at risk, exactly in the same way as uh, seasonal flu vaccine is a recognized thing that's a useful thing for people who are at risk.
Fair enough. I think the second part of her question also is, does that frequency differ from type of vaccine? So I don't know if she means type of vaccine by the Moderna versus Pfizer,
Yeah,
or
I
if
have.
she means from disease to disease. So Brooke, if you could clarify, we can come back to that.
Again, I don't have data to answer that question adequately at this time.
Okay. All right. The next question comes from, I believe it's Shilpa.
Mhm.
Is it really okay to get the second shot of the vaccine after the advised number of days? For example, if the Pfizer second dose is due after 21 days, but the shot is not available then, is it okay to take it a bit later than 21
Yes,
days?
yes, absolutely. So, what usually happens with a vaccine that works is it's administered and your body makes an immune response to that vaccine. So, all the lines of data that we read rise up. You develop an antibody response, your T cells become responsive. You may get a mild fever or what I'd call an interleukin response, such that you don't feel completely well for a couple of days. Your body's responding. Those side effects disappear typically in a very short period of time, and your response continues. And the second dose tops that up. So it goes well beyond where it ever was, and it tends to sustain over an extended period. So 2 doses, yes, very good to get that. And if there's a short delay between those, longer than is recommended, that's fine too. But again, ask your physician about this.
All right. Another question. For double masking, are two cloth masks okay if we don't have access to a surgical mask as the first of two masks?
Yes. Cover your mouth when you sneeze. I mean, that's basically it. Um, so wearing, there are some misconceptions about masks. Masks are not a panacea, but they do dramatically reduce droplet transmission. And by dramatically reducing droplet transmission from my mouth to anywhere on your body, eyes, mucous membranes, ingestion, inhalation, uh, it's a way of dramatically reducing the transmissibility of any kind of infectious particle. Masks don't have to be sterilizing N95s. I mean, an N95 isn't sterilizing. They don't need to be sterilizing masks or N95 masks because they're not filtering out viruses. They're filtering out droplets that contain viruses. So I wear a normal surgical mask or a neoprene mask whenever I'm out, and wash my hands and cover my eyes as well. But I'm overcautious.
All right. Good. Good question about the masks.
Yeah.
Um, I think it's Trelina.
Mhm.
Um, I think we talked about this a little bit earlier. Can you discuss whether the current vaccines will be effective against the new variants? I think you covered that just a little bit ago.
Yeah, and I can speak a little more broadly on it.
Mhm.
Vaccines, key messages are antigen delivery, immunostimulation. But if you make a good response against an antigen that is the wrong antigen, your vaccine by definition won't work. So if the antigen on the pathogen changes sufficiently, then by definition, your immune response won't cover it. However, functional limitation in terms of selective pressure is key. Things that need to function can only change within limited parameters. For instance, H and N, the hemagglutinin and neuraminidase antigens of influenza, are always present and do change, but they have to maintain function for the virus they are present on to be viable. Therefore, there are some soft boundaries around these things. And in the case of COVID 19, the S protein is functionally required and consequently is restrained in terms of the evolutionary variation that it can demonstrate. So I am concerned that viruses change, but that's not a surprise. That's to be anticipated. And it's great that we're able to pay attention and essentially rip the genes out of any new variant and clone it up and make a new version of a vaccine.
Cool beans. Okay. All right. Thank you for those questions. We have another one. I think it's Trineka. And if I say your name wrong, I am very sorry. I apologize because I can't hear you speak your name. So I apologize. Names are important. They say there are two different vaccines, Pfizer and Moderna. Why is it not advised to mix the vaccines? For example, if you took Pfizer as the first dose, why can't you take the Moderna as a second dose?
I don't know the answer to that question, except that a double dose mixed between two different products hasn't been tested clinically, and multiple doses of the same product have. So we always err on the side of caution. It would be difficult for me to say from a biochemical point of view why there would be any problem with that. But also, I know that there is no clinical trial data of that schedule. So I would say, yeah, it's probably fine, but best not risk it.
Well, I know also there's been some concern about how there's pregnant people, essentially, they don't always do a lot of testing. I've read that recently, and I think I can't think of which article, but
Well,
there's some concern about people who are pregnant receiving a vaccine.
yeah.
Would you like to expand on that a little bit?
Well, yes, I can. There is a requirement um, to cover a broad population in testing. And there's also a requirement to test in animals prior to any human clinical use of an experimental or other product. And it's an unfortunate reality that toxicology is tested in animals and some of them are pregnant, and that's a necessary thing. So the types of bad effect that we would expect to see in people are typically either captured at that stage or during clinical trials, and I've not seen anything to indicate a problem. Although I'm not a clinician, so I'm going to be very cautious about this answer.
Um, all right. Ashley has another question. Will the vaccine prevent you from contracting COVID?
Yes,
Yes,
most likely. Now there
most
are
likely.
no. Uh, I like to be accurate and precise. So to say yes 100% of the time would be inaccurate and imprecise. Over a population, the probability of any individual who has a functional immune system and is vaccinated, the likelihood of them catching and transmitting COVID-19 becomes vanishingly small if that vaccine is effective in that person. There are always exceptions. But if you ask me, will the incidence of COVID and COVID related morbidity and mortality in a population that is vaccinated drop relative to one that isn't? I would say absolutely yes. And
All
that's
right.
what we're going for. You know, take care of yourself and your family, but we're all part of that family.
All right. Please get the vaccine when it's available to you. If
Yeah,
you are, and your doctor recommends it.
definitely.
Jay Johnson asks, AstraZeneca recently halted their trial in Africa, stated that the vaccine was showing a 10% efficacy with the African variant. What should we expect with the Pfizer and Moderna vaccines with these anticipated variants?
I would expect them to be updated in accordance with any new variant that is becoming dominant.
How would they, just a little bit of tech here, how do they actually update a type of virus like this? How would they go about that? I know most of these are mRNA vaccines.
Yeah. The process involves essentially reverse engineering part of the S protein in whatever variant is being targeted, which is a molecular biology exercise, and then the presentation of the mRNA either in a particulate form or in a viral envelope. But that would be piggybacked on the work that's already been done. So it's not developing something from zero, it's making vaccine 2.0 effectively.
Okay. Um, let's see. Tracy McHardy says, how has the Netherlands been dealing with the pandemic in regards to regulations?
Yeah, it's been really, really good and really bad at varying times. So I've been here throughout the process. The first outbreak was treated as an inconvenience, and only after about 4 weeks did people start to take it seriously. And a lot of people got infected, and a lot of people died in that time. So at that point, there were still elderly folks on buses going to markets, and it was a nightmare. Uh, we've had two fairly firm lockdowns, which have been very effective, and the overall numbers are beginning to get under control. In terms of public response, people mask routinely. There's no issue in that. People have adapted extremely well to that. So there's very little resistance to the basic public health interventions. Um, there's actually a curfew in place at the moment, which is not rigidly enforced. It's more a sort of, yeah, it's better if you stay at home for everyone. And people are kind of pragmatic about it. And they're like, well, it seems like pretty reasonable advice, actually. And over the last week, the weather has been really bad. There's been snow and ice, and people are staying home. So it's great. The numbers are dropping based on the fact that folks aren't out and about. So any intervention will take 3 to 6 weeks to begin to show up in the numbers. I am very confident that the US numbers will begin to move in the right direction quite soon, especially since your country is now taking it seriously.
Okay. Let's see. Brooke has a question. Does the viral vector vaccine also prime our immune system to target the spike protein?
Yes.
There we go. Um, I think it's R. Ballard says, will the vaccine become an annual need to stay safe? I think we covered that. You covered that a little bit ago.
Yeah, I don't know the answer to that question, but it's a thing that we're certainly considering. And if it's actually indicated, if it's necessary, then absolutely.
All
Um,
right.
in the same way as flu is. So it's quite conceivable that this will be an ongoing thing. And I think if we can, let's vaccinate everyone and wipe this out.
There we go. And I think that covers Ashley's question. Do you foresee the vaccine being added as a routine vaccine like the flu shot? So you talked a bit about that earlier. I'm catching up on on questions. All
I
right.
saw one that popped up that was interesting. Uh, is there anything to stop rich countries buying up all of the vaccine?
Ah,
It's That's a really interesting question, and the answer is no. And I'll tell you why it doesn't really matter. Um, there are multiple companies making this, and the one that I was involved in is scaling up to the point where they're making enough for everyone with no one left out. And the other companies are doing the same. So while it takes time to produce things, the scalability of vaccine production is such that we're making enough for everyone. The the distribution and administration is really where this kind of thing falls down. If you can make 100 million or 1 billion doses of something, but you can't get it to the people who need it quickly enough, or in a way that's stable over time, so that you can distribute it all over the world, that's where things break down. But I mean, my perspective on this as a pragmatist is let's mobilize the world's military to do it. I know that would be unpopular, but that's the infrastructure that um, could really, really be effective.
All right. Good question there. Let's see. Reginald asks, can you explain how the lipid delivery mechanism of the mRNA vaccine works? And because of this new system, can this technology be used on other viruses and infectious agents?
Uh, Firstly, yes, it can, and I think has been, um, but basically, a lipid delivery system involves the generation of micelles, which spontaneously form with phospholipids and other similarly polarized molecules. And you can engineer them to encapsulate other molecules like mRNA. And a lot of this maps very closely over the prebiotic chemistry abiogenesis experiments. So once you have a phospholipid at a suitable concentration in an aqueous system, it will form micelles. There's another thing about phospholipids that's kind of cool. Some of them, and you can chemically engineer this, are immunostimulatory. And if you add additional proteins to a phospholipid matrix, you can generate what's called an ISCOM, an immunostimulating complex. It can be protein phospholipid or it can be protein saponin phospholipid. Saponin cholesterol ISCOMs are excellent vaccine adjuvants and also form neat icosahedral structures. So, I mean, that's just basic chemistry. There's no design involved. If you mix the ingredients, they will produce this kind of stuff, and it's really neat. So, I think that's the short answer. A slightly longer answer includes things like outer membrane vesicles, which are essentially natural lipid delivery systems. The classical vaccines, outer membrane vesicle vaccines for Neisseria meningitidis, are an example of this. The bacteria itself, both when grown in certain types of culture and my understanding in vivo, overproduce membrane, so it produces blebs, literally blobs of its own membrane with lots of proteins decorating them. And while they are immunologically similar to the bug, they can't cause infection because they don't have a genome. Um, and those have been used quite successfully as vaccines for that type of bacterial infection. But it's also a lipid delivery system, albeit a naturally occurring one.
It's really cool how this vaccine works. It's pretty awesome how the fact that your own cells make the antigen and it doesn't actually cause sickness. So,
Yeah.
all right. I think we had I don't know if you can answer this or not. You probably can. Dr. Roy asks, will one vaccine work better than others on organ transplant patients who are on steroids?
That's a really tough one. Um, so, organ transplant patients, by definition, um, are immunocompromised. Uh, Oftentimes, again, I'm not a clinician, so this is habit knowledge from general understanding. But a xenograft, i.e., if you have my kidney, your body will respond to it if your immune system is intact. Therefore, for it to function, your immune system has to be suppressed. Consequently, a vaccine will have a lot less to work with. And by vulnerable populations, I would include folks in this circumstance. So, some people can't be vaccinated or can be, and the vaccines won't work. And that's why the herd immunity concept is so critical. If I am immunologically vulnerable, but my circle of friends and family around me are all immune, then I'm not at risk. But if enough of them are also not immune, then I am at risk. So I get vaccinated uh, because I want to spend time with my parents again, not because I'm afraid of dying from COVID.
All right. Um, let's see. Kathy has a question.
Mhm.
And this is a pretty good question. Do you have any comments on getting the vaccine from one who has had the COVID-19 virus, so previous infection?
Yep. I've had the virus and 2 doses of vaccine. No, all the vaccine will do is top up the immune response that you've already made. And there's an upper limit to that, meaning you can't overtop the tank to the point where it causes you a problem unless you're really, really working hard at it. And a vaccine schedule won't do that.
All right. How long does, and this is from Normie, how long does it take for the vaccine to take effect and provide protection against the virus?
It's a really good point. And when my parents got their first dose, I said, this doesn't mean that you can take your mask off and party with your neighbors. Um, 5 days to a week is generally what's described, and that's a pretty good estimate. But get vaccinated and take care is the key. It's a little bit like wearing a seatbelt. You wear a seatbelt when you get into your car. It's not because you anticipate wrecking your car, but you know that car wrecks happen. So you get the vaccine, it reduces your risk. It's probably going to prevent an infection, but not immediately. So keep the seatbelt on.
We have another question.
Mhm.
There are claims that animals can carry this COVID virus, so wouldn't they have to be vaccinated as well, like humans, to control the spread of it? And that's from Tranika. I think I said it right earlier, and I don't remember how I said
Uh,
it. So thank you for that question.
yeah, it's interesting. Brief answer: if animals can carry this virus, then yes, a vaccine would be useful. On a more broader on a broader response, one of the challenges of virology is the habit that viruses have of jumping between species. So zoonotic infection is a well understood thing, and it's really cool and also really scary. Um, Controlling the animal reservoir of a virus like this has some potential, but that genie is out of the bottle now, and the transmission is from person to person primarily, or at least there's no reason to assume otherwise.
Well, thank you for that. Um, Daniel Daniel has a question, and he says, What is science behind there being 2 shots for the vaccine? Is there a difference between the ingredients or amount or something else?"
Nope, same thing. You get 2 doses of the same thing. The first induces a response that reaches level X. The second induces a response after that that tops up level X and results in extended uh, immune response in an ideal circumstance.
All right. Ashley has another question. Why does the vaccine or vaccines, why do the vaccines have side effects to the lymphatic system in reference to swollen lymph nodes?
Aha! So this is immunoanatomy. Your lymphatic system, lymph nodes are sites of immune congregation, as in sites of inflammation and areas to which cells gravitate. So, as your body responds, various things happen. Cytokines and interferons are typical, inflammation is typical. And the fluid dynamics of your body change such that certain bits expand, and your lymph nodes are one of those parts. So when you get a vaccine, if you don't have any response to it, I would be more concerned than if you do. Essentially, it's your body is responding to a challenge, to an insult that will then protect you from subsequent infection.
We have another question regarding allergies. If somebody takes only, and this is from Nima or Nima, if somebody takes only the first dose of vaccine and doesn't want to take the second dose because of developing an allergic reaction after taking the first dose, how much will it be effective?
I don't know. It depends on the individual, but looking at the kind of serology, first dose will give you some degree of protection, but about half of what you would expect with two.
So about half as much as what you would
Yeah.
expect. How do you feel about the allergic reactions? What's the mechanism, or what's causing, do you know what's causing these allergic reactions in
Well,
certain individuals?
I'd need to know what the reaction was.
Mm.
Any medical intervention, especially if you're sticking a needle in someone, can and often will have undesirable side effects. But if someone gets a vaccine and they have irritation at the site of injection, develop a low grade fever, and have achy shoulders and muscles for a day, and they call that an allergic response, that's not the case. That's your body responding to the vaccine. So it's not always pleasant, but that's a necessary function.
Let's see. Tong has a question, and this might be an ethical question.
Okay.
There's an incident in which a doctor, or the doctor, noticed the vaccines were about to be expired, but no one received it. So he gave it to his family and got punished for such action. I think that was recently in the news story. How could people just let the vaccines go expired without giving them away? Because I believe it's 6 hours they have to finish once they open a not viral, vial.
Mm-hmm.
They have 6 hours to completely use that vial before it's a ruined dose.
Yeah.
What are your thoughts on that? How could people just let the vaccines go expired,
Yeah.
or punish that particular doctor?
Well, I'd have to know the 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 facts of the case in more detail. But you said that he administered a vaccine that was almost expired to his family. Is that correct?
Um, yes, I believe that that's what the news story was. It came out a few days ago. It was featured in,
Well,
I think, the New York Times or
it,
something.
it, expiry dates and instructions for use are data driven. So if a window of use is given when you thaw a vaccine out and it's 6 hours, there is a reason for that. So going beyond it means you're administering something that is not necessarily the same product as you started with because it has changed over that period of time.
Well,
Um,
I think this doctor used up the rest of, because he couldn't find anybody else to use up the rest of the vial. So he just went and found friends and family to use it up before it expired, and that cost him his job, and he was accused of stealing.
well, that's. I
That's
think there are two questions there. The first is there a scientific issue with that product being administered? And I'd say no. And then there's a property issue and a professional conduct issue, which is a separate thing that I'm not qualified to talk about.
okay.
Uh, but if you break the rules, you know.
Yeah, I suppose that's a good ethical discussion that people might want to engage in a later time, I suppose.
I'm qualified to talk about vaccine stability and formulation. Um,
Okay.
And in use studies are part of all of the required stability studies that are a necessary function of licensure of products like this. So you study the integrity of the product over a period of time, ultimately years at -70, -20, 2 to 8 degrees, and in some instances at room temperature and in use. So, on the bench, is it the vaccine good at room temperature for the period of time that it'll be used? I'm laying that out in detail to give the impression, it's an accurate impression, that we're not guessing about these numbers. If there's a lifespan, or a stability time that's indicated, it's data driven.
All right. Johari has a question. You may have answered this question earlier, but is contracting the virus not enough to build immunity?
Yeah, but you get sick too. Um, Also, there's uh, if you get a low level infection, you may or may not mount a sufficiently strong immune response to protect yourself from subsequent infections. If you are infected and recover and then get the vaccine, great. If you are infected and recover, great. But I'd prefer people to be protected without having to get sick, is really the key.
Also, I think often people talk about, okay, it's only a 1 to 2% mortality rate, but they don't talk about the morbidities that
Yeah.
are associated with COVID-19. Maybe you could expand a little bit on how it is that so people get these morbidities.
Well, again, I'll caution that I'm not a clinician. So 1 to 2% sounds like a small number. That's 1 in 100 or 2 in 100. And it's a small number that mounts up very, very quickly when you start to talk about millions of population. And that's a lot of graves, and a lot of people have died, to my mind, unnecessarily in the US and other places. But that 1 to 2% is a misrepresentation when you look at a population of folks over 60, over 70. It's actually very, very much higher than that in those vulnerable groups. So that's more or less how I look at it.
I mean, that's a good way to kind of put it in perspective,
Yeah.
you know.
And I mean, another perspective that is stark, but it's accurate. The USA in the last year has lost more people than you did in the Second World War, and you're losing people at about 12 times the rate on a daily basis than during the war. And you're losing more on a daily basis than in the Normandy landings, the Saving Private Ryan, machine guns, bodies floating. That's every day. Um, Things will improve, but it's been quite hellish to watch.
I just living through it here too. So, yes.
So I see that we're at time now.
Yes, we're at a flat hour. How do you feel about answering maybe two or three more questions and
Let's
then we can?
let's let's do that. Yeah.
All
And
right.
if there are any other questions, if they can be funneled through you,
Okay.
for accountability and whatnot, I'll do my best to answer them.
Okay. Let's see. Aziza asks, will people of different ages or weight take different doses of the vaccine, or do we all take the same dose?
Child different, adult same.
All right. Blessed asks, has there been any recorded deaths or serious injury due to the vaccination?
Here's the thing: if you do a trial of anything with a sufficient size, size sufficient to be statistically relevant, people will die. So what I can say is, people in the clinical trial um, vaccine group, some of them died, and people in the control group who didn't have the vaccine also died. and the numbers were consistent between those two groups and not above the expected mortality for a population of that size over the time involved. What does that mean? It means that if, as some supposed news sources um, choose to, you look only at the vaccine side and say 30 odd people died in this group, that can appear to be bad. But if you look at any population of that similar size and the time period over which the study was done, and say, yeah, if you did nothing, 30 people died. If you did vaccine, the same number died. And if you did vaccine placebo, the same number died. So, in short answer, I'm not concerned about the safety of these vaccines more so than any other. And the cost benefit in terms of risk reward of being vaccinated versus contracting COVID is orders of magnitude different.
Okay. And we have one more question. This is a technical question.
Mhm.
Can you expound on the distinction between acquiring cellular entry from SARS-CoV-2 versus having an infection that is developing COVID-19, the disease? So the reason why Reginald's asking this is that some people think they are absolutely immune after having the vaccine, and they think they cannot pass on the virus because they had the vaccine. So is it still possible? I guess he's asking, is
Well,
it still possible to pass on the virus even though you've had the vaccine after
if the
having both doses?
if you have a protective immune response based on antibody response and T cell immunity, you are vanishingly unlikely to transmit the infection because the infection can't sustain itself in you. You cannot go on fire and then shed sparks because the vaccine has hosed you down at that point.
Okay. I think we have one more
Mhm.
question. Um, let's see. We've got, here it is. Is the COVID-19 virus mutating as it spreads?
Yeah.
Yes.
And the reason I can say that is because it's biological and because it's under selection pressure. And with those two things, if you propagate yourself, if you replicate yourself with variation and you're exposed to selective pressure, you are evolving. One is evolving. And that doesn't stop when those two qualities are met. And with a biological entity, they are met. And we are the environment for this evolutionary theater.
All right. I think this will be the last question that we ask and then we've hit all the questions. How amazing is that?
Great.
Diana asks, is it true that COVID originated from a bat? There is speculation on that now. This may have already been asked. Why is it so easy for viruses to jump from animals to humans?
It's a cool question. So zoonotic infection is not uncommon for bacterial and particularly for viral pathogens. The probability is that the events generating this particular viral strain involved one or two non-human species, most likely bats or pangolins, if I should say so. And those two things very rarely get together in the wild, and that's why wet markets with blood and guts and excrement all over the place are really good places for infections like this to begin. And to briefly answer a really nuanced question is, why do these things transfer from one environment, one animal species to another? One of the reasons for that is viral genomes can be segmented and in many instances are, which means it's easier for them to swap out bits of information from one to another. And many of them are also have RNA genomes, which copy themselves with less fidelity than DNA genomes. So really, it's a function of viruses being simple, quote unquote, simple enough to colonize multiple cell types or multiple species, and being mutable enough, variable enough, and transmissible enough to make that jump into other species. And the key examples for this are Ebola, which is thought to have an animal reservoir again in bats, and the most impactful, which is simian immunodeficiency virus, SIV, which is first cousin to HIV, and jumped across that species gap by blood to blood contact, as a function of hunting, killing, and eating apes. So these are known things. Also, the influenza virus has a great habit of recombining from avian species and porcine species into things that can do us harm. Again, as a function of an RNA genome and a segmented genome. So, mutability, transmissibility, and versatility of habitat are the key issues.
Well, I think that's a good rounded out answer. Do you have any last comments, Dr. Boulton? I want to thank you again for coming back out and doing another Q&A. You're such an invaluable resource, and it was so successful last time, and this is always an ever evolving virus, and so it's really important we stay up to par on the current information. So thank you. And any last comments before we wrap up?
Well, I'm really grateful for the opportunity to present and to speak and to answer these questions, and it's fun for me. It would also be fun if we weren't experiencing a public health crisis. So I'm not deriving any morbid pleasure from that, but I'm impressed with the standard of the questions, the angles that the questioners are taking. I think we've discussed some really interesting and very valuable information here, and I'd encourage people to consider some of the things that have been discussed and seek some answers to them. But don't worry, we'll totally get through this, and hopefully learn a great deal from it.
Well, thank you so much. We're all in this together, aren't we? We've got to work together and promote the correct information. So thank you for the work that you do, Dr. Boulton, and thank you again for coming out. And we will be posting this somewhere on the Southwest site, so feel free to share it with anyone else who needs additional information about COVID-19, and hopefully this talk will be helpful.
Perfect. Many thanks to everyone in the audience and to you for arranging this. Consider me a resource, and I will sign off for now.
Okay, thank you, Dr. Boulton.